Bismillahirrahmanirrahim. Welcome to my channel. In this video, I will be explaining the resting membrane potential in the excitable cells. The concentration of ions in the extracellular and the intracellular fluids is largely balanced with a net neutral charge. However, a slight difference in charge occurs right at the membrane surface, both internally and externally. The membrane potential is a difference of charge across the membrane measured in millivolts. The standard is to compare the inside of the cell relative to the outside. So the membrane potential is a value representing the charge on the intracellular side of the membrane, assuming the outside being zero. It is the difference in this very limited region that has all the power in the neurons and the muscle cells to generate electrical signals, including action potential. In excitable cells, the membrane potential recorded at resting conditions is called as the resting membrane potential. The resting membrane potential exists because of a small buildup of negative ions in the cytosol along the inside of the membrane and an equal buildup of positive ions in the extracellular fluid along the outside surface of the membrane. Such a separation of positive and negative electrical charges is a form of potential energy which is measured in millivolts. The greater the difference in charge across the membrane, the larger the membrane potential. The exact value measured for the resting membrane potential varies between cells, but minus 70 millivolts is most commonly used value for most neurons. The minus sign indicates that the inside of the cell is negative relative to the outside. A cell that exhibits a membrane potential is said to be polarized. A major factor that contributes to the origin of the resting membrane potential is the unequal distribution of various ions in the extracellular fluid and the cytosol. Extracellular fluid is rich in sodium and chloride ions. In cytosol, however, the main cation is potassium. This causes diffusion of ions down their concentration gradients. Secondly, the plasma membrane typically has more potassium ion leak channels than the sodium ion leak channels. Hence, the number of potassium ions that diffuse out of the cell into the ECF is greater than the number of sodium ions that diffuse from the ECF into the cell. As more and more positive potassium ions exit the cell, the inside of the membrane becomes increasingly negative, and the outside of the membrane becomes increasingly positive. Most anions inside the cell are phosphates. They cannot follow the potassium ion out of the cell because they are attached to non-diffusible molecules such as ATP and large proteins. Membrane permeability to sodium is very low because there are only a few sodium leak channels. Nevertheless, sodium ions do slowly diffuse inward down their concentration gradient. Left unchecked, such inward leakage of sodium ions would eventually destabilize the resting membrane potential. The outward potassium ion leak and inward sodium ion leak are offset by the sodium potassium ATPase pumps. These pumps help maintain the resting membrane potential by pumping out sodium and bringing in potassium ion. Recall that the sodium potassium ATPases expel 3 sodium ions for each 2 potassium ions imported. Since these pumps remove more positive charges from the cell than they bring into the cell, they are called electrogenic, which means they contribute to the negativity of the resting membrane potential. Their total contribution, however, is very small, only -3 millivolts of the total -70 millivolts resting membrane potential in a typical neuron. Resting membrane potential is the steady state of the cell. This is a dynamic process that is balanced by ion leakage and ion pumping. Without any outside influence, this state will not change. Stimuli originating from neighboring cells or external environment can bring changes in the resting membrane potential. Alterations in membrane potential are very important since these are used as electrical signals in the nervous system. The alterations in the resting membrane potential are of two types. The first type is called the local potential, also known as the graded potential. The local potential is used an electrical signal over a short distance. The most important example of local potential is the electrical signal moving from dendrites to the cell body and eventually to the axon. The second and more familiar type of electrical signal is called the action potential, also known as the nerve impulse. This is used as an electrical signal over a long distance all along the length of axons of the neurons. The example is the nerve impulse generated in the axon, which travels to the end of the axon and releases neurotransmitter at the subsynapse. Thanks for watching and listening. Please do share this video with your friends and colleagues. If you like this video, please subscribe to this channel and watch more videos.